tidal group. So those are all things you want to know. In addition, like I said, to the lipid component, there are proteins that are embedded. Some proteins can span the entire membrane, so they can go from one side of the membrane to the other. That spanning process might involve an alpha helix moving its way through that space. It can involve a lot of different things. So there are a lot of pictures in your book. You can Google integral membrane protein, peripheral membrane protein, and see images of those things. I encourage you to do so. One way that the protein can be associated with the surface of the membrane, that hydrophilic part, with peripheral membrane proteins, is just by having, what are those things? If you don't know what I mean by when I do this, I don't know what you did. So when I do this, what am I needing you to think of? Weak interactions, whether it's hydrophobic, ionic, hydrogen bonding, hydrophobic. So that's the kind of forces that are going to govern the interaction between a protein and the surface of a membrane. So if you had to guess of those different which ones do you think are going to be involved? Based on what you know about what this part of the membrane is. Remember, it's the hydrophilic part. So what kind of interactions would we be looking at between the protein and the membrane surface? Okay, there are 4 of them. We just said what they were, and now I'm asking you, which of those 4 would you predict to be involved in the interaction of a protein with the membrane surface? All you have to do is say one of those 4 things that make sense. Maybe hydrogen bonding. Now, that assumes that you know chemically what this structure is. So make sure you look at that. Make sure you realize that that involves a phosphate group that's going to have a negative charge. Make sure you realize that there's going to be something else esterified to that phosphate group, like a serine or a threonine or anything that could have a hydroxyl group. It doesn't have to be an amino acid. It can be something like an ethanolamine. So any of those things could be part of that head group. So in addition to hydrogen bonds, what other kind of interactions do you think might be involved in the interaction of a protein with the surface of a membrane?
Hydrophobic interactions.
We're talking about peripheral membrane proteins. We're not talking about proteins that span the membrane. Ionic. All right, I said that. Good. So any kind of polar interactions that could occur would be ones that would stabilize that interaction of a peripheral membrane protein with the surface of a membrane. How do you think you could disrupt that interaction? Again, what kind of things do you think could disrupt hydrogen bonding or ionic interactions, these weak interactions? Maybe change the pH. If you change the pH, that can result in a functional group that was normally in an ionized state becoming protonated if you decrease the pH. And if that happens and there was an ionic interaction occurring and you no longer have ions, then what happens to the ionic interaction? It's gone. You can't have it. You have to have things that are ionized. Remember, these interactions are reversible. That doesn't mean that that protein just binds to the surface and then just willy-nilly moves away. Those interactions can be pretty extensive and can be pretty stable, meaning that that protein that's associated with the surface isn't just going to easily come off. But if pH changes happen, if salt concentrations change, what kind of interactions would changes in salt concentration disrupt? What do you think? Ionic, because the salt contains ions. And if you dump a bunch more salt in there, those positive and negatively charged ions could interfere with the positive and negative interaction that occurs between the protein and the membrane surface. And that's actually the kind of thing that a biochemist actually does. It's using that information about what we know about those interactions in a process. For example, maybe what you're interested in as a biochemist is to determine whether a protein is integrally associated with the membrane or peripherally associated with the membrane. So what you can do is you can isolate that structure that has the membrane and the protein associated with each other. And if you do something like a relatively mild change in pH, or you change the salt concentration of the buffer that system is in, and then you see that the protein and the membrane are no longer associated with each other, then what kind of association was it? Okay, two ways that the protein can be associated with the membrane, either integrally or peripherally. If you do something like a pH change or a salt concentration change, and you see that interaction be disrupted, then what kind of interaction was occurring between the protein and the membrane? Peripheral. It's a peripheral protein. Because that kind of interaction is not going to allow an integral membrane protein to be plucked out of that membrane. What kind of treatment do you think you'd need to make an integral membrane protein dissociate from the rest of the membrane? What do you do to dissociate grease from your hands? You use detergent. Do you use only detergent?
Hot water.
And water. You need both. So one of the ways that is used to dissociate an integral membrane protein from a membrane is to treat that system with some kind of detergent. And again, you do an experiment where you have a system that has a protein associated with the membrane, and you treat it with mild salt concentrations or pH changes, and the protein does not dissociate from the membrane. treat that same system with a detergent, and then that causes it to dissociate, then you have an idea, well, that seems like that's probably an integral membrane protein. Another way that you could get an idea of whether or not a protein is integrally associated with the membrane or peripherally associated with the membrane by using things you already know in this class is by examining the linear sequence of amino acids. Now remember how I said in some instances, one of the ways that a protein can span the membrane is by having an alpha helical structure be involved in that spanning. So, think about the direction that the R groups are pointing in that alpha helix. If you don't remember that, write it down. They are pointing radially outward from, or perpendicular to the axis of that helix. Now think about what kind of environment is this within that lipid bilayer. Are you writing these questions down one by one? Because that's the reasoning process that you need to do. So what kind of environment exists in that lipid bilayer? Hydrophobic. So now, next question, what kind of amino acids would you be expected to find in that alpha helix? Residues that have non-polar R groups. So I think the thickness of a typical membrane, somebody Google it. Hey Google, what's the thickness of a typical membrane? I think it's 45 angstroms. 45 angstroms? Anybody else get that? Every single one of you have a phone, so I know more than one of you can Google it. Is that what you found? Say again. What'd you get? Say it. 5 to 10 nanometers. Are those two about the same? Remember what an angstrom is? An angstrom is 10 to the minus 10 meters. So that would be 45 times 10 to the minus 10 meters. Or 4.5 times 10 to the minus 9 meters. 10 to the minus 9 meters is a nanometer. So 4.5 nanometers, is that in line with this? Yeah, okay. You should know those kind of things, folks. That isn't just something you learned in Gen Chem and then you forget forever. The reason it's important is, if a membrane is about 45 angstroms thick, how many amino acid residues in an alpha helix are needed to span that 45 angstrom distance? How many amino acid residues in an alpha helix are needed to span that 45 angstrom distance? So now what do you have to remember about that to do that problem? So, the main thing that you have to know is what is the rise that each amino acid residue contributes when it is in an alpha helix. Do you remember that? Essentially, think of that spiral staircase, and you're on one step, and you step up another staircase, the step. It's that distance between the two steps.
3.6.
Anybody remember?
3.6. It's 3.6.
Angstrom?
I thought 5.4 was for the 100 for the bond day.
3.6 is
Okay, there's a lot of numbers that are thrown around here, they're gonna get us close. So, the one thing that you know, the 3.6 is 3.6 amino acid residues per turn.
I've been telling my friend.
Okay, so it takes 3 amino acid residues, basically 3.6 steps, that you go from this location up or down so that you're directly above or below where you were. Okay, what other numbers were you saying?
5.4 angstroms.
Okay, that's different.
Is that distance?
Yes.
Okay, so when we do this, if we consider the distance between here and here, that was 5.4 angstroms, wasn't it? So now, and that's 5.4 angstroms per turn. When I say turn, I mean going from one location, so you're directly above or below where you were before. So now, given that information, answer the question, what is the rise that each amino acid residue provides when it appears as a residue in an alpha helix? You have all the information you need right here, including the ability to use dimensional analysis.
I did it the other way, so I put 33.6 on the top of it. Oh, that's not what it's on the bottom. Yeah, I know, but like it doesn't. When you think about it,
Well,
I'm okay on it. Okay. It would look see, because like even if it was 3.6 rate per turn.
So what we're interested in is how many angstroms per amino acid. If you're one amino acid residue appearing in an alpha helix, what distance does that move that helix along that axis? So that's the units we're interested in, right? So to get those units from these units, what do you have to do? You have 5.4 angstroms per turn times 1 turn. It's 3.6 residues, amino acid residues. Turns divides out, so you have angstroms per amino acid residue. So what's the answer? 1.5 angstroms. Now, let's go back to our question. How many amino acids, I think you already have this written down in your notes, it should be right before we did this, how many amino acid residues would need to be present in an alpha helix if that alpha helix spanned the typical membrane?
30.
How many?
30
How'd you get that? 30 what?
residue.
30 amino acid residues. So the way you get that is, you know, if it's 1.5 angstroms per amino acid, and you know you got 45 angstroms, 45 divided by 1.5, ends up giving you the number of angstrom, or the number of amino acids that it would take to move that 45 angstrom distance. So about 30 amino acids. About 30 amino acid residues. How many turns would that alpha helix make as it spans the membrane? How many turns would that alpha helix make as it spans the membrane?
Oh, no, wait. I'm thinking wrong. Yeah, you're right. So 8.6.
So about 8 turns. So in a book, if you see a cartoon picture of an alpha helix going through a membrane, go ahead and count the number of turns to see if they're being accurate or if it is just really a bad cartoon. Okay. So now, this whole discussion is the foundation for you being able to answer this question. How could examining the primary structure of a protein provide evidence that the particular protein is an integral membrane protein? How could examining the primary sequence of a protein provide evidence of whether or not that particular protein is an integral membrane protein? Can we talk to each other? I'm gonna go 2.5 minutes to talk about this because this is complicated.
Are you okay?
So we see the amino acids and you can see the properties of them, and if they're hydrophobic or if they're non-polar, then you would know that they're in the hydrophobic region, and if they're
We already went through this question. Like, we're doing it again, but in different words. So it's like, we're learning so much. Yeah.
Yes. You think of integrals in the If I'm looking at the primary sequence and I don't see any,
Because it has to be from non-clear state.
I should see about 30 amino acids length sequence of non-polar amino acids, basically.
Where that?
I don't see that.
So this picture here might help your discussion. Here's a membrane. Here's a membrane, integral membrane protein that spans. Here's the N-terminus of the protein, here's the C-terminus. I just randomly did that. This could have easily been the N-terminus, and this could have been the C-terminus. So, you come up with an answer? What'd you come up with?
Me?
Yep.
if you see the primary structure, you can observe the properties of its you can see its amino acids, and you know that if it's a non-polar, for example, string of amino acids that are non-polar, you can assume that it's in the hydrophobic region. So you know that it's integrated.
So that's pretty good.
Am I rambling?
No, that's fine. When anything you said that was wrong. If you look at that sequence, and you're looking at the nature of each amino acid residue, whether it's polar, charged, uncharged, hydrophobic, those are the characteristics that you would be looking at. And if you see a string of, say, 30 amino acids that are all hydrophobic in a row, and then you look and you see maybe interspersed or maybe a bunch of hydrophilic, then that can make you think, well, 30 amino acids, wait a minute, that's exactly the number of amino acids that would occur, or not exactly, approximately the number of amino acids that would occur in an alpha helix that was spanning the membrane. Wait a minute, I know that that's a hydrophobic environment, so I would expect all of those amino acid residues to have hydrophobic R groups. So if it had that, maybe it is an integral membrane protein. Any questions on that reasoning? So that kind of thought process ties together a lot of what we have done so far this semester, hasn't it? How about if you saw a primary sequence that was like this? The blue represents a stretch of mostly hydrophobic amino acid residues. Given this analysis of the primary sequence, predict the topology of that protein in a membrane. You know what I mean by predict the topology? So you just did that a little bit ago when you said if you saw a sequence of about 30 primarily hydrophobic residues, you predicted that the topology of that particular protein would be that it spans the membrane one time. So, answer that question about this. Given this analysis that you have multiple, mostly hydrophobic regions, and how many do we have there? 1, 2, 3, 4, 5, 6.
Asking if
Let's
the
say
fundraiser,
7.
if the fundraiser will pay the full cost or if they'll half the cost. What's that one? Are we paying it? Or is it fundraising?
So predict the topology. What could you say to answer that question? To answer the question over here, we predicted the topology by saying that that protein spans the membrane one time. So how would you say the answer to that question? The protein's topology is that it does what? it spans the membrane 7 times. I mean, how complicated is that logical leap? If you have one section that's hydrophobic and it spans the membrane 1 time, and you have a protein that has 7 hydrophobic regions, you would expect it to span the membrane 7 times. Draw that. Even more explicitly, draw that so that the N-terminus in your cartoon is shown on the cytosolic side of the membrane. Label your picture so that the N-terminus is on the cytosolic side of the membrane. It's nothing special about how you draw it, it's how you label it. Is that what you drew? You see that that answers that question? So make sure you got all that written down, including the question and the reasoning. I'll step out of the way so you can write it. That kind of motif is not an unusual way that integral membrane proteins are associated. There are a lot of integral membrane proteins that have this 7 topology. Now, does that mean all of them do? No, some can just have one. In fact, some integral membrane proteins can be associated with just one of the leaflets. It doesn't even have that particular part of the protein go into the other leaflet. So any possible arrangement can happen with proteins being associated with the membrane. What would you predict about the nature of the amino acids that are in this region of the protein? Would you predict, how do you answer that question, first of all?
Polarity, charge.
Yeah, just polarity, charge. So what would you predict? A lot of non-polar. Now, does that mean there can never be any polar functional groups in that region? No, they could be there. It's just that most of them would be non-polar, so that you could have these hydrophobic interactions, van der Waals interactions, governing that association between the acyl side chains and the R groups in the residues of the protein, or the amino acids that are in that protein. Questions about any of that? Your book doesn't talk in detail about that in this chapter. So that's something I talked pretty extensively about it. I think it's important for you to know that. So, other ways that proteins can be associated with membranes. Sometimes the proteins are What anchored means is, for example, that a fatty acid like a palmitoyl, palmitate is a fatty acid. It's how many carbons are in palmitate? Hey Google, how many carbons are in palmitic acid? Can you find it?
This is the top result.
Leave me hanging. 16. So you can have one, two, three, four, five, six, seven, eight, 9, 10, 11, 12, 13, 14, 15, 16. You can have that carboxylate functional group covalently bound to an R group in an amino acid residue. The common way to do that is to have that bound to a nitrogen. Do any amino acid R groups have nitrogens in them? Which one you think of? Lysine. So if there is a lysine residue and that palmitoyl group is attached to that, that palmitoyl group can be embedded in the membrane, and therefore the rest of the protein is going to be anchored to the membrane. Because that palmitoyl group is embedded in the membrane and is covalently bound to an R group on the protein, so that is a way to anchor a protein to the membrane. Okay. Any questions about any of that? So let's talk about what membranes are doing. One of the things that membranes provide is a barrier between one three-dimensional space and another three-dimensional space. That's not the only thing that membranes do. They do so much more. There's a lot more they can do. But that property of providing barriers between three-dimensional spaces is that specific organelles that occur within eukaryotic cells can have a much different chemical environment than other three-dimensional spaces within that cell. So that's one of the things that the membrane can do. Another thing that a membrane can do is it provides a place for proteins to occur so that those proteins can do their job. One of the major jobs of proteins that are associated with membranes is to signal something happening outside that membrane to tell the chemistry and everything that's happening inside the membrane that needs to happen. That's basically called signaling. If you have a protein on the surface of a cell that's integrally associated or even bound by a palmitoyl group, and some other small molecule binds to that protein, that binding can result in the change of conformation of that particular protein. We've seen that repeatedly, haven't we? That binding of some ligand to a protein can cause a conformational change to that protein. Well, that conformational change that happens to that protein can result in something happening inside the cell. That kind of thing is called signal transduction. If there's some need to have some chemical process occur inside the cell because something happened outside the cell, you need to get that message across. You can't just have somebody pick up their cell phone and call from the outside of the cell to the inside of the cell and say, hey, we need you to ramp up this metabolic process. The way the outside talks to the inside is through some chemical process. There's the signal transduction. Another job that proteins can have when they're associated with the membrane is in allowing other molecules to cross the membrane. There are a number of different scenarios that occur with these type of proteins. And there's actually a figure 5.4 on page 144 in your textbook. 5.4 on page 144. That describes different functions that various integral membrane proteins can have. So in addition to that signal transduction process, they might act as a transporter. They might act as a pump to actually move things energetically across the membrane. They can act as a channel. So all of those examples are given here in that figure. Some vocabulary that you need to know are what a uniporter is. Uniporter. What do you suppose a uniporter would do?
One-way direction.
Transport one thing in one direction. How about a symporter? What a symporter does?
Transport two molecules in the same direction.
Okay, transports two molecules and they're both going in the same direction. How about an antiporter? What do you suppose that does?
Transport in the opposite direction.
Transports one molecule across the membrane in one direction and at the same time transporting another molecule across the membrane in the opposite direction. So they also talk about how some of those proteins can be regulated. One way that they can be regulated is by having a ligand bind to that integral membrane protein. Sometimes what happens is a ligand can bind to the protein and that opens up a channel. Sometimes what can happen is a difference in ion or a difference in charge across the membrane can cause that channel to open through that protein. There are some instances when that process of transporting a molecule across the membrane does not require any additional input of energy. If the process does not require any additional input of energy, that kind of process is called a passive transport process. If there is the need for an input of energy in some form, what do you suppose that kind of transport is called? Active transport. So those are just different ways that the proteins can function. Another thing that they mention in the book at this point is a process called osmosis. So do you remember what osmosis is? is the movement of water molecules across a semi-permeable membrane in the direction of lower concentration of some solute to higher concentration of that solute. And when you look at that later and you think, oh no, I think you got it mixed up, then it should be from higher to lower, or from lower to higher, duck season or rabbit season. The way I've said it is correct. I'm sure it's correct. The water goes from a lower concentration to a higher concentration. And think about it. What the system wants to do is equalize the concentrations across the membrane. The way to do that is to dilute the higher concentration so that that higher concentration decreases and then reaches an equal concentration to the lower concentration. The only way to achieve that in osmosis is to have the water move. The ions don't move. And then they also show a cartoon picture of, in figure 5.5, of the protein that's anchored in the membrane. Another possibility is that in addition to it being anchored through a fatty acid, sometimes you can have even a covalent bond to the head group, to some part of the head group. So those are different ways that proteins can be associated with membranes. As far as the makeup of membranes, remember I said how the main lipid component of membranes are glycerophospholipids. And glycerophospholipids are just a class of molecules, because we can have something like phosphatidylcholine, or phosphatidylethanolamine, or phosphatidylserine. There's a lot of different groups that can be esterified to that second position of the phosphate group. So the proportions of those different membranes depends upon the cell, it depends upon where that membrane occurs, if that membrane is part of the mitochondria, if it's part of the nucleus, if it's part of the cytosol. Even from one leaflet to another, the cell regulates the makeup of the lipids that are in that particular membrane. Don't worry about memorizing what those proportions are. Just realize that that's something that is very closely controlled in the cell, from membrane to membrane. Another process that can occur in membranes is something called fusion of membranes. What do you suppose fusion of membranes involves? Two membranes coming together. Another process is called budding. What do you suppose budding involves? Exactly. So in your book they show a picture. I don't think it's a very good picture, so maybe my description will be just as good or just as confusing. So remember, these membranes are three-dimensional structures. And one thing that can happen is that you can have a membrane, think of it as just being a surface, and you can have imagine grabbing a hold of some of that membrane and grabbing it and twisting it. And then pulling off that piece that you twisted. The thing that you pulled off is gonna be surrounded by a lipid bilayer. So hopefully that's clear. It's surrounded by a lipid bilayer. It's not a monolayer, it's still a lipid bilayer. There's a volume inside that thing that you twisted off of there, that you butted off of the membrane. What are the contents inside that? So if you have, here I'm just gonna draw the membrane as a line, but make sure you realize what I'm talking about there is a lipid bilayer. And we squeeze this together, so this membrane, and I guess I'll show it in a couple of steps, eventually pinches off, and eventually you have that. So let's say this is the inside, this is the outside of the membrane. Let's say it's a cell membrane. So, pathologically, what's inside that vesicle? Are the things inside that vesicle the things that were here or the things that were here? It's what was inside the cell. Okay? If the reverse process occurs, where you have a structure like this bind to the membrane and then go through a fusion with the membrane, where do the contents of the interior of that vesicle end up? Inside the cell. And that's actually a process whereby a cell can make something inside itself and get that stuff outside. By having the stuff made inside, having a vesicle pinch off, and the contents inside there are the contents of whatever was inside the cell. And then when it does that, that vesicle is then outside the cell. Then perhaps that vesicle can fuse with another membrane and deliver those contents to another cell. Maybe those contents can be dispersed because the membrane gets broken down. So those are things that can happen within a cell that are important with what happens in a membrane. And it's actually a really complex process that I drew here. The one thing you need to think about is what molecules are involved in this budding and fusion process. Given what you know about membranes at this point, what molecules are going to be involved in this budding and fusion process? Proteins involved. What else? And phospholipids or membrane lipids. Those are part of the membrane, and those are very specifically going to be working with each other to accomplish this very complex process. So far, so good. So the next section in your textbook is about a process called signal transduction, which I already alluded to. There is a figure, 5.5 in your textbook, that describes that process. And what they're showing in that figure is a membrane, and they're showing something happen, and let's say that membrane is a cell membrane. So you have an outside of the cell and inside of the cell. And you have something happen on the outside of the cell. That something could be that a molecule like insulin comes along. insulin come from? Where was the insulin made? In the pancreas. How did it get to a cell down here in my leg? Did it just get a shot and inject it there? Some people have to do that, but the normal process, the insulin is produced in the pancreas. Then where does it go to? There's a blood vessel from the pancreas to the liver, okay? And then there's lots of blood vessels going from the liver to everywhere else. So eventually, that insulin molecule can find its way to a cell all throughout your body. And the reason that insulin was released from the pancreas is because perhaps you just ate 5 pieces of pecan pie. Why would you do that?
Probably good.
Or maybe you just ate a bunch of arepa dulce, or maduros, or what's another good thing? Or a coquito.
Coquito.
Or a bunch of Snickers bars. Whatever it is you ate, what would you eat?
Rubbing coconut.
Rice pudding. Oh man, I gotta tell you about this.
Sounds good.
It's so good.
There is a place in New York City. Did you ever go to New York City? Have you heard of New York City? There are ways to get in there. I don't necessarily have to have a car.
What's in New York City?
But there is a place called Rice to Riches.
Right.
That's a play on the term rags to riches.
Have you heard this one?
Rice to
I wonder
riches.
if he's heard this one.
Only rice pudding. They have like 40 different flavors of rice pudding. I'm just getting hungry thinking. It is so good. So, you should treat yourself at some point. First of all, if you don't know what rice pudding is, you're really missing out. So you were eating rice pudding. A lot of sugar, a lot of carbohydrates, which is more sugar, is present in this. You eat a bunch of that, you're going to have a bunch of sugar circulating in your circulatory system. So that sugar needs to get inside the cells. It's not doing any good if it's outside the cells.
Aren't you like
The way that gets accomplished is the insulin is released, the insulin circulates through your body, the insulin binds to a receptor on the surface of cells, and ultimately that results in that sugar being transported through a transporter into the cells. It's a little bit more complex than that, but you get the idea. There are a lot of processes like that where something happens and a signal is released from somewhere in the body, and that signaling molecule needs to have a way to say, hey, this is what just happened. You need to do something inside the cell. That's a signal transduction process. Your book shows a figure where there are 2 different general signal transduction processes. One is, or both of them involve some ligand binding to a receptor on the surface of the cell, and that receptor is a protein. One of those processes involves the ligand binding, and that ligand binding results in another small molecule. Remember, the ligand is a relatively small molecule. It's not this great big protein. That the ligand binding results in another molecule being produced inside the cell that then binds to other cellular components, telling them, hey, you gotta do something. That idea is the production of something called a second messenger. The first messenger is the ligand. It might be something like insulin, or it might be some other hormone. The second messenger is another molecule that gets produced inside the cell to cause some other process to occur in the cell. Yeah, the second messenger is going to be a ligand to some other molecule inside the cell. The second messenger is not a ligand to the original receptor. Another general process involves a ligand binding to a receptor on the surface of the cell, and then that resulting in a conformational change, which can then end up causing other conformational changes to occur, essentially without a second messenger needing to be produced. Again, that picture is on page 149, figure 5.8, looks like. So a particular one that I want you to know about is one, it's called the PKA signaling pathway. The PKA signaling pathway. I'm gonna have to ask you people to not sit next to each other anymore, because you're continually talking, you're continually distracting me, and if you're distracting me, I'm pretty sure you're distracting the people around you. If you decide to keep talking, the next time that happens, I'm going to ask you to leave. Look at me. Look at me. Do you three understand that?
Okay, we got it.
Good. You didn't get it last week when I had to do this, but I hope you got it now. I apologize to the rest of you that had to see me discipline college-level students. I shouldn't have to do that. The PKA signaling pathway involves the binding of a ligand. It can be something like a hormone. The particular example in your textbook is, it talks about the hormone epinephrine. So when that ligand binds to the receptor, there are proteins inside the cell that are associated with that receptor. The protein, in this case, is something called a G protein. G proteins are a multi-subunit protein. One subunit is called the G protein. And you have another subunit, actually another couple of subunits, that are associated with that G protein. What happens, and remember, those are physically touching the receptor molecule on the inside of the cell. So you have the receptor molecule that spans the membrane. You have this protein complex that is touching that part of the protein. When a ligand binds, that causes conformational changes to happen at the interface between the G protein and the receptor. This is on the outside of the cell, this is on the inside of the cell. When that conformational change happens to the receptor, there is an exchange of GDP for GTP. in the inactive state, there is a GDP that is bound to one of the subunits. When the conformational change happens, that causes that subunit to release a GDP and bind a GTP. Is that clear? This protein doesn't have GTP bound. There's not any hydrolysis of a GTP to produce GDP. There's an actual exchange of a GDP ligand to a GTP ligand. When that occurs, that causes the other parts, the other subunits of that G protein to dissociate. The way they distinguish this in your textbook is, for that G protein, there's an alpha, beta, and gamma subunit. There's 3 subunits. The alpha subunit is the one that involves the GDP to GTP exchange. So when those beta and gamma subunits dissociate, oh, by the way, remember we learned about how proteins are integral membrane proteins, or how they're associated? Both the alpha and gamma. Both the alpha and gamma subunits, let me draw the gamma subunit up here a little bigger. Both of those are associated with the inner leaflet of the membrane. So that's how they stay close to the membrane, is because they have a component that's embedded in the membrane. So when this exchange occurs, the beta and the gamma subunits diffuse away from the alpha subunit, still associated with the membrane, and the gamma subunit causes the activation of an enzyme that's also associated with the membrane, causes the activation of another membrane-associated enzyme called adenylate cyclase. What adenylate cyclase is an enzyme that catalyzes the conversion of ATP to cyclic AMP plus pyrophosphate, the 2 phosphates. So 2 of the phosphates get removed from ATP, so you have AMP. Instead of just having AMP, you have cyclic AMP. In particular, it is 3', 5' cyclic AMP. Remember, the prime refers to the position of the carbons in the ribose component of that nucleotide. This cyclic AMP serves as a second messenger. So let's recap the whole process. Something happens, that a hormone is released, that hormone circulates, binds to a receptor on the surface of a cell, that causes this G protein coupled receptor to be activated. By the way, that's what this is called. This is a G protein coupled receptor. That G protein coupled receptor is activated. The process that I described ensues, resulting in the activation of adenylate cyclase. Adenylate cyclase is the enzyme that catalyzes the production of the second messenger. The second messenger, cyclic AMP. Now remember, the other thing about this is it just takes one signal molecule, one first signal, one hormone, to bind, and that will result in the production of a lot of cyclic AMP. Is that clear? Because that's an enzyme. And an enzyme is going to catalyze a reaction. If there's plenty of ATP, you're going to get plenty of production of cyclic AMP. So that second messenger that gets produced what that cyclic AMP will do is it will bind to an enzyme called protein kinase A. Protein kinase A, protein kinase A in an inactive state consists of two catalytically active subunits and 4 regulatory subunits. Cyclic AMP binds to the regulatory subunits. So 4 cyclic AMPs are going to bind to the regulatory subunits, and what do you suppose that's going to cause the regulatory subunits to do? Undergo a conformational change. And when they undergo a conformational change, when they bind that cyclic AMP, everything dissociates. And then what do you suppose happens with these? Here, those catalytically active sites are in an inactive state. Once they're unbound from the regulatory subunits, you have these active. What do those enzymes catalyze? It's a protein kinase, remember? So what do you suppose that does? It catalyzes the ATP-dependent phosphorylation of some other protein. Okay. We'll continue talking about these things next week, or Thursday. Please look at the figures in the textbook. There are videos you can watch about this. I extended